stuff. So good morning and good afternoon to everyone who's joined us today. Welcome to LabKey's Learning Academy, covering LabKey Server for research studies today. My name is Jason Ledley. I am the Director of Operations based in the UK. And on the screen then, joining me from Seattle to deliver today's session is Steve Hansen. And Steve is our Director of User Education. Good morning, Steve.
Good morning, Jason. Good morning, everybody.
Okay, so it's great to have so many of you join us today. Thank you for doing so. And if you did join us when we ran a training webinar series earlier this year in April, thank you and welcome back. It's good to have you with us once again. And it's actually due to the success of that webinar series in April and the feedback we received that we've decided now to go forward with these additional, more frequent training sessions. And these are intended to be more structured, are more application specific, focused on real world use case applications of LabKey Server. So that's the kind of training that you've asked us to cover. So we're delighted to be able to meet that request. Steve, if you go to the next slide for me, please, that would be great. So as most of you will know, LabKey Server is a highly flexible and configurable toolkit. We often refer to it as a toolkit, and it's configured specifically to solve a very wide range of different research data challenges across the research environment. There are literally countless different ways the platform can be used. All of these are real world examples. You go to the next slide there, Steve. One of the most advantageous applications, though, one of the key strengths of LabKey Server is around the longitudinal study data management area, where LabKey is particularly good at the integration, the management, analysis, and the secure sharing of study data, clinical study data, where uniquely, in many ways, LabKey can tie together demographic data, phenotypic data, clinical data, laboratory data, specimens, and numerous different assay data at the participant level. And so today we'll be looking at LabKey for research study data. Next slide there, Steve. Firstly, by way of an agenda with an overview, really, of what we mean by research study data for LabKey, before looking at some real-world examples. Steve will also then actually build in real time a portal from scratch. And then we'll end with looking at some of the tools that are available in LabKey Server to allow users to publish and share research data. If you have any questions, please do raise your hand or use the chat facility within Zoom. I will be facilitating and monitoring that. We'll try and get through as many of those as we can during today's session and break occasionally to do some Q&A. But for those we don't get through, we will be collating those questions and reaching back out to you with those answers sometime soon. Okay, so with that, let's dive right in and let me hand you over to Steve Hansen, our Director of User Education. Over to you, Steve.
Great, thank you so much, Jason. I'm gonna turn off my video just so it's a little less distracting. All right, well, as Jason said, we're gonna do a mix of kind of feature overviews today and also real world building of something out. We're going to really start from scratch and show you how to put together a portal using the study features. But first, I thought let's get oriented around LabKey Server and its tools and kind of get a sense of what their breadth is and what use cases they're really for. So a quick overview around LabKey study features. Why use LabKey for your study data? And when I say study data, I really mean a pretty broad cross-section of research. Sometimes people call these observational studies, longitudinal studies, research studies. There's other cohort studies. LabKey is really useful in all these cases. It's really optimized for that whole broad range of work that you could be doing. And so we kind of lump them all together, sort of use those terms all as synonyms for this presentation inside the server. first thing that comes to mind, why would you use LabKey Study for this kind of stuff? Well, you can just do, it's a great tool for managing the data on a day-to-day basis. You know, these kind of studies take sometimes years to complete, and the data trickles in bit by bit, and you need a place to put it, centralize it, latch it all together, you know, do quality control on it. There's lots of process around this kind of data. And so you really need a place to, a central place for the data to collect and funnel, get all your eyes around it, collaborate around it, discuss it, decide where you're going with it, what you want to do with it. So as just a day-to-day tool, it's a great tool. Also, anytime you are tracking subjects over time, the LabKey Study features are a great choice. All of these studies, this kind of research work involves ultimately tracking performance of some subject or some participant over time. You want to be able to, because ultimately what you're doing here is you often have a control group and then another group that you have somehow changed, or you've maybe given them a treatment or something. And you want to be able to track those two groups against one another over time and see their performance changes. Also, anytime you have lots of different kinds of data, LabKey Study is a great option. Oftentimes, research studies have data that come from all kinds of different sources. We call it heterogeneous data sources. You may have questionnaires. You may have medical histories, clinical observational data sets, laboratory data that came out of some assay or some experiment, specimens, etc. All of these different kinds of data sets have different shapes. They all need to be hooked together in some way and analyzed together. LabKey's really optimized for that kind of a problem. It's also a great place, again, to just collaborate on data together and to divide and conquer your work around this data. Somebody on your team might be assigned to quality control. Somebody on the team might be assigned to data modeling. Somebody else is a specialist in analyzing the data and building visualizations around it. Again, LabKey's got all those features in place. Also, LabKey is about presenting your research to the world or to some more elite team, like some other research group, or just the researchers in your own group. LabKey has lots of features around building that relationship between your data and your audience, whoever it might be, whether it's a big audience or a small audience. And it's also great at protecting PHI and PII data. And we're gonna look at that at the end. How can you make sure that you're sharing just that sliver of data that you want to share and holding back that data you don't want to share, like people's names, people's addresses, etc. So these are all great reasons to use LabKey. The LabKey study features, or study module, we'll call them. Well, what is a LabKey study? Well, what it does is it is a active data parsing environment. It has concepts already built into it. Namely, I'm thinking of 4 main concepts. There's a concept of a subject, and the subjects of your research can be pretty broad. LabKey's not just for animal models like primates or humans. We've had people put trees inside the study module. We've also, obviously we've had lots of people use it for animal models, and for organisms, but people have even placed molecules as the central subject of the study module. Think big when you're thinking about the study module. It can really handle a lot of cases. The second dimension, the second concept that's built into the study module is time. Your data can be encoded either as dates, calendar dates, or as some kind of number. But you need to provide the study module some way to capture time, because that's ultimately what you're going to be doing here. Is you're going to be tracking your subjects over some time period. And so these are the time slices that you have as part of your research. There's also a concept of specimen inside the study module. And then cohorts also. These are the groupings that you placed your subjects under. Anytime you put data into the study module, LabKey is going to look for those concepts. It's going to look for the data that corresponds to those concepts, and it's going to parse it, it's going to align it, and it's going to pre-analyze it. And that's going to make your final analyses a lot easier. This pre-analysis, it allows you to, and pre-integration, it allows you to really browse all the data as a single connected whole. It allows you to do things like build queries and tables and visualizations that cross all the different data sets that you may have brought into your study. But what I mean by that is it's going to allow you to take a part of your specimen data and a part of your laboratory data and a part of your clinical data and view it all together as a single table and do those cross data set analyses. It allows you to do sophisticated querying of the data, like we call it the slice and dice side of analysis, where you can just look at one facet of the data, et cetera. And it also lets you monitor really how your study is progressing. Are you 50% done? Are you 10% done? Are you finally done and ready to present it? To make this a little more concrete, I think let's take a look at some real world examples out there. And first of all, let's take a look at something called the Open Research portal. This is a LabKey site that involves about 6 or 7 different teams. And they're all doing research on the Zika virus and the coronavirus. This is a particular portal, a particular study that these groups have presented to us, and it's called Zika virus 23. This is a very generic use of the LabKey study module. They haven't really done much to customize its look and feel. This is pretty much the default presentation for a research effort. And I think it's just looking at this is a great way to sort of learn the anatomy of a LabKey study. First off, notice these tabs. Each one of these tabs presents a different dashboard or different facet of their research. There's an overview tab, and this will provide a bird's eye view of the data. And also, it gives you an opportunity to present the rationale for your research. Like, what's your research all about? What are you trying to discover? What questions are you asking? What are your hypotheses? And the Zika team here is presenting us with that rationale, with a breakdown of the participants, et cetera, and also visualizations and the actual summary results of their research. We'll take a look at that in a minute. But there's also a participants tab here. This is a breakdown of each individual subject in the study. And if I drill into one of these participants, this is a code number for one of those participants, I get a profile of the data that's just for this participant. there's a third tab here, clinical and assay data, and this is a summary of all of the base data sets in this study and all of the artifacts that they've built on top of those base data sets. And I mean by that, custom grids and visualizations. Here are their visualizations. If I hover over these links, I can get a preview of them. And here are their base data sets. These are all just tables that they've assembled and presented here. And then finally, there's a specimen tab. And this particular effort doesn't really have any specimens, so this is a relatively empty dashboard. They probably could have just taken that tab off. If we drill a little bit further down into this overview tab, take a look at this sentence right here. This is really interesting. It says Zika virus 023, that's the name of this study, tracks data in 34 data sets over 513 time points. Data is presented for 4 participants. You might think that this was a sentence that was written by a human as a summary of the study, but in fact, it is not. This is a machine-generated sentence. It is, if you like, you could think of this as the first report, auto-generated report that we've encountered in our examination of the study module. In fact, all of these panels, like the participants tab, the participants dashboard, was machine generated. These participant data profiles were machine generated. These are the products of LabKey's parsing of the data and of its concepts around the data. You didn't have to hand assemble this. All you have to do is throw the data into this active parsing environment, and LabKey gives you all of this stuff for free. It doesn't give you the visualizations for free. You have to build these yourself. But all this parsing and joining is something that you don't have to work for in LabKey. It's not going to write your hypotheses either, but you get the idea. Let's also drill down a little bit more here. These panels in LabKey, these are called web parts. And Jason mentioned earlier that LabKey is a giant toolkit. That's right. And these panels are the tools. There's hundreds of these panels inside the server. And you work with LabKey by surfacing these panels and creating dashboards. These particular dashboards are just the default dashboards, but you have total control over these dashboards. You can customize them the way you want. You could get rid of these panels if you don't like them and bring in new ones, or customize the panels that you're offered. And that's what they've done here. They've added this text to this text panel to give us a sense of what they're doing. They've added these links to highlight the main parts of their research, the main results of their research, etc. Notice this visualization panel. This is a chart panel or web part. And one of the things that's also notable about LabKey is how much control it offers the end user. Here I can turn on and off these elements of the chart if I want. I can also view the underlying data for this chart and then hop back to the visualization itself. I can do the same here. This one is, frankly, there are some kind of broken things inside of this portal. It's a very raw portal. The Open Research Portal really is a mix of finished and unfinished research. And so you'll see some sort of stuff that's halfway done as you surf through this portal. And I really recommend you do so. This is publicly available. There's lots more to see than just this one portal I'm looking at. But here is, if I go down to the bottom of this page, here is a LabKey data grid. And I just wanted to stress to people how much power you have as an end user when you're working with a data grid in LabKey. I'm just looking at this grid. It's a necropsy tissue report. It tells you the viral count for all these different tissues inside the subjects. Just one question I have for myself is, well, what kind of tissues are involved here? Well, you can find out by clicking and calling up a pie chart for yourself. It gives you a better idea of the variety of different tissues that might be in this grid. You can also ask, well, what tissue had the most viral copies in it? Well, you could do that. You can find that out for yourself by just sorting this viral copies column by descending order. Which tissue had the most viral copies in it? The mesenteric lymph nodes, as you would expect. Which had the least? Well, looks like the eye, aqueous humor in the eye, the kidneys, the lungs, they didn't have a lot of virus in them. Also, another thing you can do with LabKey, these researchers have invited us to actually download this data. You can actually just export this grid and bring it into your own analysis systems. Now I have an Excel file of this grid, and I can, if I have my own systems, I can analyze it there. I can also modify this grid if I want. Notice there's a customized grid panel here. I can open this up, and I can add a column if I want, like the day here. That might be useful for my analyses. And there's even other data sets that are behind the scenes here that I can pull into this grid. So if there's data in this activated NK cells grid, maybe some of these have data that's relevant. I'm going to just pull these columns in. I don't even know what's going to happen, but let's take a look at that grid now. There's not much data in it. Wasn't a very successful exploration of the data, but as we'll see when we build our own portal, we're going to make all these same kind of moves and as part of our broader analysis of the data. I did see there's some questions in the chat here. Anything, Jason, I should address before I keep going?
There's a couple I've answered directly, but there is one.
Yeah.
We understand that to join data, LabKey requires a participant ID and some concept of time or a scheduled event, as you mentioned at the beginning. Yeah. But how could LabKey handle the integration of data that may not have a time or an event-based element to it?
You could use a lot of different features for that. I would just think of SQL queries would be a great way to do it. And really, depending on the shape of your data, you could also use something called a lookup in LabKey. That is a graphical way to join data from different grids. It is true that one kind of data integration requires a participant ID and a time point column. But by no means do all forms of integration require that. That's really just that's kind of the classic study way of joining data. But there's lots of options for you, a lot of broader options for integrating data as well. Good question, actually. Anything else there, Jason? I see them coming in. Else, yeah.
There's another one here. Yeah, so that last question actually was referring to study. So if you're using a demographic data set, you know, without a time primary key.
Oh, yeah. Yes, that is also possible within the study. If you just have a participant ID, you can actually pull that into other data sets as well. And we'll take a look at that. When we actually build something out, I think it'll give you a more concrete sense of how that's done.
Okay, cool. Thank you.
Great. There's nothing else. I'm just going to keep going. There's lots to see here. So anyways, this is the kind of your basic introduction to LabKey UI. There's tabs or dashboards that you can configure. There's panels that you can surface to provide different presentations and analyses of the data. And there's also this menu over here. This is the folder menu. This gives you a broad sense of everything that's in this server. Each one of these links is a different research portal. And they're arranged like a folder tree. They have nodes that you can open and close and surf around just like any kind of folder tree you've seen a million times. Notice the portal we were just looking at, Zika virus 023, is part of a much broader series of studies, all of which have similarities, but they all have slightly different hypotheses. They use different subjects, although some of the subjects are shared across all of these different research efforts. And so this Zika virus experimental team has a whole bunch to show. I really recommend you surf around and take a look at this portal. Even though some of it's pretty rough, it's a pretty wild research portal in that respect. There's some of it's completed, some of it isn't completed. Notice I was just surfing around in an area that looks like it's kind of under construction. Its parent folder is called Do Not Use Until Further Notice, but I still recommend you look around in there. Another portal that is really interesting is the ITN's, or Immune Tolerance Network's trial share portal. This is a much more polished portal. This is really intended for presenting journal publication ready data. And this is from a group called the Immune Tolerance Network. This is a large consortium of researchers who all work together on autoimmune diseases. You need a login to get into this site, but you can just ask for one from them. They'll give you one, and then you'll be allowed in to see their data. When you first enter their portal, you're going to be presented with this sort of getting started tutorial screen. I really recommend you follow this through and take a look at it. It's a great introduction to their portal. I'm going to skip it. I'm just going to give you my introduction to their portal, but I recommend you don't do that and do it for yourself. Notice their UI is a little different. They've really customized their UI, and they have the familiar tabs over here, but they're different. They have different names, etc. And instead of a folder menu, they've presented each one of their studies as a different tile here. This is their unique UI that they've created for themselves. Again, I just want to stress to you, you're not stuck with the vanilla UI in LabKey. You can do something like this if you want. And you can either just surf around these studies and click into them, just get a summary, look for something interesting, and surf in. But they also present you with different ways to filter the studies down to just the ones you're interested in. Like, what if you just want to look at the allergy studies? That will filter these panels down to a smaller group. Or suppose you have a study in mind, you can actually just do a text search for it. I know that this Leap On study by Gideon Lack is a really interesting one, so I'm just going to surf into it and take a look at it. First thing they present to you is these are the journals which have published this data. And there's a number of different journals or different actual publications that are on top of this data. And I think this one is a particularly interesting one. It's about peanut allergies. It's about, and they've challenged their subjects with peanut proteins to sort of see their reactions. Here is their list of base data sets. The base data sets are down here, and these are those data grids, just like we were looking at previously. They have so much data that they've done some styling here to really shrink down everything into these tiny little text fonts, just because it's so rich. But again, we have all the same control over these grids that we did back at the Open Research Portal. If I want to get a sense of, well, what are the different treatment groups that they've used? Well, there's two different treatment groups. If I want to get a sense of, oops, excuse me. If I want to, filter or sort any of these grids, I can. They've also let me export this data. So if I want to bring this into my own analysis system, I can. I'm also able to just visualize this data on my own terms if I want. I can go into the visualization wizard here and start playing around. Like if I put age on the, no, let's put something more interesting there. If I put participant ID on the x-axis and age on the y-axis, I can get a breakdown of what kind of folks are in this study. Yeah, well, something went terribly wrong there. Let me try that again. I'm going to do a box plot of age on the y-axis and participant ID on the x-axis. Well, something's gone wrong there. In any case, let me back up and look at the visualizations they've provided me. Here's the visualizations that they are going to give me. And this one is an interactive R report. This is the rendering of that R report. And here are the parameters. I can change them if I want. If I just want to look at the males, I can re-render the report. And it'll take a minute, but it'll redraw it for me, etc. And I can keep going. Notice they've also provided, as part of transparency, the actual R code behind this report. So I can evaluate that, make sure it makes sense, they're not making any analysis mistakes in it, etc. So that's just a few of the ways that real researchers are using this study module. Notice they have a very large study here of 640 participants, 556 are officially enrolled, all of these different cohorts and groupings that I can surf around and look at. All of these are different data profiles again of individual participants, slightly different UI, but it's the same spirit, the same kind of experience that they're providing us. I saw lots of questions coming in. Anything I should speak to, Jason?
We're okay for right now, Steve.
Okay, great. Well, let's keep going here. I've also, these slides, I'm going to make these slides available to you. But I think let's actually build something. How'd they do that? How did those two teams or those two sites get put together? And really the rest of this presentation is really a practicum about how to work with the study features. First of all, I'm going to start out by looking at some fake data that I created. This is a set of Excel files, comma-separated files, tab-separated files that I've collected on some subjects to study HIV. Again, this is data that I invented. It's not real data, but it's supposed to it's really inspired by real events, if you like, in HIV research. I have here 6 different data sets. The first one is a demographics data set. It's a tab-separated file, just a text file. And it gives me little profiles of 12 different participants. There's a lab results file, and this is a blood count assay. It gives me the CD4 counts, CD4 white blood cell counts, the total lymphocyte counts, and a hemoglobin level. There's also a medical history. And, oh, are you guys, I got paused somehow. Can you hear me, Jason?
Yes, we can. That's fine. Yeah.
Can you see me moving around here?
No.
Okay, it says my screen sharing is paused. I think somebody might have pressed share by mistake. I'm gonna steal it back. Okay, I'm back. I think I'm back here. There's also a medical history data set here. And this one is kind of interesting. Notice it's got that radioactive data in it. I've got full names here. I've got street addresses, got dates of birth. Obviously, I don't want to share this data. And we're going to end today by showing you how to hold this data back and how to protect this PHI data. And then it's got other typical things in a research study. It's got a physical exam. Every time my subjects came in, I took their weight, their temperature, their pulse, etc. And then I've got these viral load data sets. I've actually used two different methods to get a viral load count, something called NASBA, and then a PCR style count. So right now, I don't have anything other than these Excel spreadsheets or these tabular file formats. The question here is, well, how do I get these into a study? Well, let's do that next. I also have a wish list for this data. Like, what do I want to do with this data? Well, I think the most important thing I want to do is I want to be able to compare the different treatments that I've provided these participants. I've provided some of these folks with antiviral treatments. Some of them I haven't. And I want to be able to compare them, like which treatment is more effective, which treatment, what's the health metrics for these folks. And I'm going to use two metrics. I'm going to count their white blood cells, their CD4 white blood cells, and I'm going to count their viral loads as my metrics for treatment effectiveness. I also want to be able to chart those things over time and get a sense of, as the CD4 levels go up and down. I also want to be able to compare different columns from the different date spreadsheets. I want to pull in, the weight column from one and the CD4 column from another and put them side by side and do an analysis. I also want to be able to group my subjects by cohorts, and those cohorts are going to be the treatments. I also want to assign QC states to that data. I want to, if a data is suspect or if a vial has been contaminated or something like that, I want to be able to mark that data as suspect. And then ultimately, I want to publish this data online, just like ITN and Open Research Portal, but I want to do it in a de-identified fashion. I don't want to give people's full names out on the web, obviously. So that's what I want to do with this data, and let's do it right now. First, I'm going to go to my own LabKey site. And you can actually go to this site. It's called academy.lkpoc.labkey.com. It's kind of an ugly URL. And I'm going to put it in the chat, actually, which I think you'll be able to see. And this is to all, I'm going to send this to all attendees. So here's that link. You'll be able to see this site. And again, this is a very vanilla, typical site. Here is how you navigate that site. There's about 6 different folders in it here. And you're only able to see 2 of them. There's one called Public Studies and one called Home. I invite you to go and surf around what you can see. If you open up Public Studies, you'll see there's 2 studies that I've made public here, Hansen 004 and Hansen 005. I'm going to be working in this private area that you can't see, called Private Studies. I have a whole set of studies in here, 5 of them that I've already created. I'm going to create a 6th one. And then later, I'm going to make it public. But right now, I'm going to start a new research effort. I'm going to do that by creating a new folder or a new study portal. And I'm going to give it a name. I'm going to call it Hansen 006. And I'm going to activate the study tools by clicking this study module button right here. This is what I'm saying here is make a new piece of real estate on the server called Hansen 006 and pull in the study tools. That's all I'm saying here. I need to decide on the permissions for this study, and I can either choose that this portal is just for me privately or that it inherits the same security as the parent folder, which is private right now. And here it is. Here is an empty study portal. There's not much to see yet. There's just a button or two buttons. One of them is to create a new study. Another one is to import a study. If I have one already existing, I could use this import a study. I'm going to create a brand new study. And this is the first set of questions LabKey is going to ask me about my study. It's actually asking me about what's your data look like? The first panel here is asking me where is your participant data inside of your data? Because LabKey is going to need to know where the participant data is, where the time point data is, and where the specimen data is, and able to do that parsing we talked about earlier or looked at earlier. I'm going to take all the defaults and say my data is in the participant ID column. My subject data is in a column called participant ID. I'm going to have LabKey look for assigned visits as time points in the data. And what I'm telling LabKey here is look for numbers. I've encoded my time points as numbers, not as dates. But I could pick dates as well, but I'm going to go with the numbers style. And for specimens, we're not really going to do much with specimens today, but we're just going to take all the defaults here. And with that, LabKey is ready to start parsing my data. Notice it's given me these default dashboards. It's looking a lot like the Open Research Portal already. Here is the Overview dashboard or tab, the Participants tab. There's nothing in it, but it's just kind of waiting expectantly for me to start throwing data into it. There's also a tab that you haven't seen yet, the Manage tab. This is for administrators of the study. When you publish a study, this isn't going to be shown. This is just for manipulating the data from behind the scenes. Notice this sentence right here. Here's that generated sentence. It says Hansen 006 tracks data in 0 data sets, 0 visits, 0 participants. Well, let's get some data in there and start doing something. Any chat questions I should look at, Jason, before keep going?
No questions, Steve.
Okay,
I'm just looking at your battery level and your power for your laptop.
Oh, okay. I think I'm okay.
All right. Okay.
We got 2 hours left.
Okay. Good.
Hey. So first off, I'm going to blow through a lot of user interface here. There's too much to learn. I'm just going to tell you briefly what I'm doing as I go. I'm going to create a new data set. What this means is I'm going to import one of those spreadsheets into LabKey and turn it into a data grid. I'm going to start out with the demographics data set. And I'm going to have LabKey infer the fields of this data set for me. And I can just do that by dragging the demographics data set right on top here of the fields. And this is LabKey has just parsed this data set. It's detected there's a participant ID field, a sequence num field. That's what I've called my time points, sequence num. There's a date field if I want to use it, etc. And etc. This right here is I need to help LabKey decide. I need to help LabKey to point at where's my participant column and where's my visit column or time point column. And with this, I've imported my first data set. Things start to come alive in the UI now. LabKey now has detected that there's 1 data set, 1 time point or visit, and 12 participants. It's already started to parse and build those participant profiles. Here's the participant profile for PT 101. Here is my growing set of data sets and artifacts, which I haven't built out any yet. Let's do that again. Things get more interesting when you add a second data set. Let's do physical exam. Let's do it the same way. I'm just going to drag and drop physical exam into the portal. It's parsed it. I'm going to tell LabKey where my time point data is because it couldn't figure it out for itself. Got two data sets now. Notice LabKey detected that there was lots of time points in that last data set that came in. There are no more participants. There's just 12 participants. Things are starting to get more interesting. Actually, by the way, there is a quicker way to do this. Instead of going through this bit by bit, I thought I'd just back up and show you a faster way to do this. There's actually a way to just have LabKey infer all these data sets in one big swoop using something called a file watcher. And this is a data detecting mechanism I can use. And I'm just going to set it up right here. I'm going to say, make a file detector that detects data set data. And I need to give this detector a name. I'm going to call this the Dataset Inference Detector. I'm going to turn this on. And I'm going to tell LabKey where to look for it. By putting a dot here, I'm saying look in the file roots for this folder. Now, if I go into, there's a little bit of UI I haven't surfaced here called the file browser. If I drag any data into this file browser, LabKey's just going to parse it all and put it in my study. So I have 4 remaining data sets here. And I'm just going to drag them into the file browser. And LabKey, well, I'll do it one by one, I guess, should just do this for me all automatically. Let's see what happened. Sure enough, it detected all of my data sets, all 6 of them, and it just put it all together for me. LabKey's now finished all that pre-processing and parsing. Each one of these profiles has really gotten quite detailed. And great, I'm ready to start analyzing this data. That was a lot already that I just showed you. I know a lot went by really fast. Any questions I should touch on, Jason, before we keep going?
No, I think we're good, Steve. Thank you.
Great. Well, let's keep going here. I'm going to skip some of these slides because we're running a little bit slower than I thought. Well, now that LabKey has analyzed or pre-analyzed that data, one of the first things you can do is immediately start joining grids and building hybrid grids. And let's actually skip right to the end. One of the things that I would love to know is I'd love to get a sense of how people's health fares, how their vitals are faring compared to their white blood cell counts. Here I've got people's weight and temperature and blood pressure recorded. I would love to see the white blood cell, one of the white blood cell columns inside this grid, and then start building visualizations on top of those. So what I'm going to do is I'm going to go into my grid customization panels. And notice there's a node here called datasets. If I open this, there are the other datasets in this study. I know that the CD4 and white blood cell columns are in this lab results dataset. I'm just going to grab them and pull them over. Let's grab CD4 and lymphocytes. I'm going to resave this grid. Here they are. You can see now I have my first hybrid grid. These are all native columns to the physical exam grid, but these two at the end, they're from a different data set. but I can build visualizations on these all the same. Here's just a graphical sense of how that's done in LabKey. So it would be really interesting to compare CD4 with weight, just to get a sense if there's a pattern there. So I'm going to go into the Visualization Center. And let's put let's make a scatter plot of CD4. Let's put it on the x-axis and weight on the y-axis. And there's kind of a slant here in this data. I'm going to refine it a little bit more. Maybe this will make the slant a little bit more apparent. Let's make the width 600 and the height 350. Might show a little better. Sure enough, I mean, there's something there. If I put a trend line on that, I would see something. Let's just save this for later. This is the weight CD4 comparison chart. And let's do that again. Let's go back to my hybrid grid and let's do the same thing with temperature. Let's make another scatter plot with CD4 on the x-axis and temperature on the y-axis. And sure enough, there's something going on here. Let's refine the layout a little bit. Let's make it 600 by 350 pixels. There's something there as well, and let's save this as temperature compared with CD4 levels. Notice now I have two artifacts. I've got two visualizations. Those appear in this growing list of data resources here. Here are my base data sets. Here are those visualizations that I just put. Well, let's get a little bit more detail built into this by configuring our cohorts, our groupings. You can again do that automatically with the data. All you need to do is just tell LabKey where the cohorts are. And they're already baked into my data. I'm telling LabKey here, look in the demographics data set and look at the treatment group column. I could pick any of these as cohort columns. I could pick HIV, the status of the infection, but I'm going to pick the treatment group. LabKey will automatically assign the cohorts. It's detected 4 different groupings. There's 2 treatments, ETR and DRV, and then there's a no treatment group and then an HIV negative group. It's assigned my participants accordingly. And this will create another dimension that I can layer into my visualization. So let's take a look at weight again, the weight chart. Let's edit it and let's include the cohort information here by changing the color of those data points. Sure enough, things are starting to look a little bit more interesting here. It looks like the red group, this HIV negative group, they have sort of higher weights. The other groups that are HIV positive, treated or not, they have depressed weights, which is maybe what we would expect. Let's resave this. And let's also do the same to the temperature group. Let's add cohorts as a colorant here. Again, it looks like folks who are HIV positive have these elevated temperatures, which again is maybe something you would expect. So I'm starting to get some insight into this data just real quickly with using these standard tools. Let's keep going. I'm gonna skip some of these. I also thought, I did notice that in our group, there's a fair amount of data scientists that are here. And I thought we could do an R report instead of using the LabKey's, instead of using LabKey's native tools, you can also use R to build reports. So I'm going to create an R report on top of physical exam. And I have some R code here that I can just copy and paste into this R builder. This is LabKey's native R designer. And what you do is you pick a data set, which I did, physical exam. And what I'm telling LabKey here is apply this R code to the physical exam data set. If I click the report tab, it's going to run the report for me. And lo and behold, I have my first R report on top of my data. Let's save that, make it another artifact. Oh, and let's also make this available to all users. Great. So I've already got 3 meaningful charts on top of my data, one of them being an R report.
Steve, while we're doing, Steve, just quickly, on the bottom of the screen that you're sharing, there's a black bar that we're seeing on the screen.
Is it? Yeah, I've had problems with this before. What this is this is the Zoom
control bar.
The Zoom thing, and it's sort of always in the way. Is this better or worse?
In the way. I just wonder if the 3 dots on the right-hand side, is there a way up? Then that's better.
Is that a little better?
That's a little better.
Maybe over here. I've never really quite been able to fix it.
You can drag it down.
I'm going to try to pull it down. Is that any better?
It was better when you just had it before.
Okay. How about that?
Yeah, that's better.
Okay, sorry. It's just a drag. I've never been able to really master that, and let's maybe try to figure it out. Let's do a post game afterwards and try to fix that. You can also, by the way, do the same thing in RStudio. There's a way to point RStudio at a live server. I want to keep going. I was hoping to demo that, but let's keep going. I think we're running out of time. I want to jump to the end here of my analysis and do my main result. My main question for my research is, how do the different treatments compare against one another, especially around CD4 levels? So let's go back to our data. Let's go to the lab results data set, and let's chart the CD4 levels for different treatments over time. And you do that in LabKey using something called the time chart. And a time chart lets you pick a column, and it puts time on the x-axis and time on the y-axis. Now, one of the things interesting here, why don't I see my columns? Like, I don't have anything to pick here. Well, sometimes you need to do a little housekeeping with time charts. You actually need to tell LabKey which columns to include in the time chart wizard. So I'm going to go into my lab results data designer. I'm going to go back to the definition of this data set, and I'm going to go into my fields here, and I want to work with the CD4 field and tell LabKey this is a measure that you should utilize. You might as well do it for the others as well. Let's also make lymphocytes a measure, something that's included in that time chart wizard. Save these off. Go back to the lab results data set and re-enter that time chart wizard. Now those columns are available for selection. Let's get a visualization on CD4s. And now you can see I've got my time or visit points are on the x-axis, CD4 levels are on the y-axis, and it's starting to put together the chart for me. It's not quite what I want though. Where are my groupings? I just have participants here. I want to actually re-render this chart as groupings, and you can do that by saying, I don't want to render this chart in terms of participants. I want to render it in terms of my groups, my cohorts. Now I've really got somewhere. Here is a chart that's telling me how the different treatments compare against one another. People with no treatment, or excuse me, people who are HIV negative are across the top here. They have relatively normal CD4 levels. The other folks have had some kind of challenge by the virus early on, but then as the treatment progressed, their CD4 levels get relatively normal, or at least get elevated again. That's a pretty important piece of information. I'm going to save this. I'm going to call it the main result. CD4, treatments compared. Let's call this our main result. It's so important. Let's snap it onto our overview tab. To do that, I'm going to enter something called admin mode. This is where this allows me to snap on panels, snap off panels, etc. And I'm going to add a new panel here called the report panel. And there it is. These are all my reports, but I'm going to pick the main result. And I'm starting to build a custom dashboard here. So how long did that take me? 10 minutes, 15 minutes to get this far? I've already got something I want to show to the world. Let's start to do that. Questions before I keep going and change mode?
There is one on the time plotted there.
Yeah.
There's can the time plot with groups include error bars on the time points?
LabKey does not come with native error bars. However, there's a couple of ways to do it. If you are JavaScript proficient, you can actually get JavaScript control of this chart. It's actually just a JavaScript chart. And right here, notice there's a couple of export options. I can export this as a PDF or an image, PNG, but there's also export a script. What that really means is get the script and modify it. Here it is. If you know your JavaScript and your D3 and Raphael packages, you could put error bars here, you could put a trend line in here, etc. Also, you could get error bars and trend lines and whatnot in R. So if you were to re-render this in R, you could do the same. But no, we do not have error bars natively in the graphical UI. Other questions?
You just mentioned R there, Steve.
Yeah.
And the question is, can you install custom R packages into LabKey?
Yes. Yes, you absolutely can. And again, you can even use RStudio where you have also control over your custom packages. So yes. Anything else, Jason, before we keep going here? Okay, great. Well, let's keep going here. Maybe we'll get back to do some more R stuff at the end if we have time. Another one of my items on my wish list was to do some quality control work. So let's see what that's like. First thing I need to do is just decide on what quality control states I want to have. So here is where, this is a panel where I can decide what kind of states my data can be in. I can say I'm going to just define 3 states here. I'm going to say data that's passed review, data that's been rejected by review, and data that's not been reviewed yet. And now these are 3 states that I can paint onto my data. In fact, I probably should have done this first because I can define default QC states on import. Right now, there's sort of none of my data has any states, but what I really should have done was define these earlier, and when I imported the data set, they're all by default not yet reviewed. But we'll do that next time when we do it all over again. But now I have those states available for me. So if I go into physical exam and I decide that some of these items are suspect and they got rejected by review, I can now say these 4 data points are bad for some reason. There's just some suspect reporting here, so I'm going to reject these. These data points are gone. I don't want to include them in my analyses. Or also this viral load data set. Suppose there's some problem or some questions about these, or there's something not quite right about them. I could say, these are still in review. They're just not yet reviewed or something. They're still under review. Don't include them in your analyses yet. And now LabKey is actually aware of this, and it'll give you a way to kind of go back and find those QC states. Here is the biggest summary of my research that I have so far. It gives you, it's sort of the big chessboard, all of the data points and all of the data sets. But I can filter this view by QC state. So which data points have not been reviewed yet? Oh, it's these 4 data points from viral load. And which data points have been rejected? Oh, it's these 4 data points from physical exam, et cetera. So again, this is a great tool for getting your study progress, getting a sense of where it's at, and getting a sense of which data should be included and which shouldn't. Well, we've got about 20 minutes left. I want to quickly show you how to publish this. And there's a number of ways to do it. I'm going to use something called the Publish Study Wizard. And what that is for, it's for publicly releasing the data. It's for supporting publications. And it gives you, really what publishing a study does is it gives you a way to make a precision clone of your study. But it lets you, edit out certain parts of the study as you go, or scramble parts of the study, like you can de-identify the data in it. So let's take a look at that. I actually got a couple of methods. I don't know if I'm going to be able to get to the second one, but let's do this study, Publish a Study Wizard, and get you a sense of how it works. The Publish a Study Wizard, what it does is it basically walks you through all of the items in your study and asks you, do you want to include it or not? And what this is going to do is it's going to create a clone of the study, which I can make public to the world. I'm going to rename this clone Hansen 06 Public. And as you go through the wizard, it's just this, it's going to ask you bit by bit, what do you want to include? I'm going to include all the data sets. I'm going to include all the visits or time points. I'm going to include the specimens, even though I don't have any. I'm going to include all of that. I don't have any lists. I don't have any grid views. I'm going to include all my reports. I'm going to include all this other stuff. And here's the really interesting page, the last one. This lets you de-identify the participant IDs. This will give me a scrambled version of the participant IDs. This will shift the dates of all of the dates in my study will now become fictionalized, if you like. What this does is it actually offsets the dates for a given participant by a random number of days between 1 and 365 days. But it shifts each participant by the same number of days. And that will preserve its analytical worth, its scientific worth, even though it's fictionalized. If I had any clinic names, I could mask those. And so let's finish this and let it run. And as it's running, it's completed. And let's take a look at what it did to the data. First off, notice it created a new version of the study right here as a subfolder. And if we go and look at the date, say, of demographics, notice all these dates have changed. In the original study, all of these dates were February 2nd, 2020. Everybody started out, everyone in my study in the real world started out on February 2nd, 2020. But in my new scrambled de-identified version of the study, here at public, I gotta find it, just a minute. I now have new fictional dates. Also notice the participant IDs have been scrambled. It's no longer PT101, PT102. LabKey's assigned these random 6-digit numbers. So I'm kind of halfway there to getting a de-identified study, but I still have this problem data set, medical history. What do I do about this? Full name, street address, yikes. I don't want to show this stuff. How do I handle it? Well, I'm going to first of all tell LabKey that these columns are special. I'm going to tell LabKey those are PHI columns. They contain protected health information. Let's go and mark them as so. So I'm going to go into the definition designer for medical history. I'm going to open up the field designer. And this full name field, I'm going to tell LabKey, this isn't just any old field. This is a full PHI field. Treat it special. Same with street address. Surely that is a PHI field. And notice there's different levels of PHI. There's full and limited. That's in the actual specification. Dates also are PHI, but I'm just going to use those two fields just to make the point. Now I'm going to go and do that wizard all over again. I'm going to make another version of the study that's more protected. Actually, you know what? I take it back. I am going to redo the public one. And it's going to let me go through the wizard all over again. Well, actually, I take it back. Did I do something? No, I'm in the right place. I'm sorry. I'm going to do this all over again. I'm going to start a new one. Let's call it 007, just to give it a unique name. Again, I'm going to just do everything as we go. It takes a bit of time to get through this wizard. I'm going to, again, shift everything, but here's what I skipped last time. I'm going to tell LabKey, don't include the PHA columns in this version of the study. And let's let that run and build this out. Now I've got two public versions. 006, I would never want to make public. It's got flaws. But 007, it does all the same things. It scrambles the dates, and it also has excluded those radioactive columns, the full name and street address. This is something I'm comfortable making public. I do have date of birth in it still, but in any case, let's make it public. And I can do that just by moving this folder into the public area. And I can do that with the drag and drop here. I'm going to take Hansen 007 and move it into the public area. I'm going to drop it. And now I have my first published study. If you go to this site, you'll see it right here, and you can surf around it and do all the things you can do with a public study. There is another method to do it. That is sort of the clone a study way to do it. There is also a way to do this by actually working in place to exclude PHI data. And I can do that just by actually, if I wanted to publish Hansen 006 just right out of this folder, I could have a similar effect by excluding those PHI columns using another instance, but we're a little bit short on time. I figured we should take questions for a minute. Maybe we'll have time to show you that second method of doing it, but it's much the same kind of study publication effect. Anything I should, any good questions, Jason?
Yeah, there is one. Yeah, you just published 2 studies, 6 and 7. Question, or if you were to do what you just did twice, would the randomized participant ID be the same in both cases, or is it random in both cases?
I think it would re-randomize them.
Yeah,
I'm not totally positive. I think I'd probably have to go through it again, but yeah. I think it would just re-randomize them. But I'm not 100% confident about that. Anything else?
No, that's good for now, Steve.
Okay, well, let's, I thought I would maybe round this out by, I know there's a lot of data scientists out there, and I think it's really great. I think the coolest demo I didn't get to do was the RStudio demo. I have RStudio installed on my local machine, and I thought I would open it up. And how can I point this? Because I think we've seen a lot of questions here about how far the visualization engines go in LabKey, and they're pretty good, but they're not as good as R. And so people love to work with R. It's the ultra powerful, one of the ultra powerful visualization engines. You can actually point your own R instance at a LabKey site. And I'm going to go to a more complete site called, I want to go to this Explore. This is on labkey.org. This is the finished version of the data that of the study I'm trying to build out. This is, if you gave me another hour, I'd be able to build this out. But I do want to go to the physical exam grid here and build an R report on this using RStudio. What I'm going to do is I'm going to go to the export button. Now I can export as Excel. I can export this grid as a text file, but notice I can also export, in quotes, as a script call. There's a whole bunch of different scripts I could write to export this grid. I'm going to pick R. And what this will do is this will create a little R snippet that I can just copy and paste into RStudio. I'm just going to copy this out, go over to RStudio, and grab that data. Sure enough, this grid that just popped into this panel, this is live data from that labkey.org site. It just queried that data. And now that RStudio is aware of it, it has code completion. All of that data is packed into an object called labkey.data. That's the data frame that I just grabbed. And I can pull in columns and start doing plots on it. So if I want to get that blood pressure plot to re-render, sure enough, this is again live. This is live data on that site that RStudio is able to see and build upon. So I definitely wanted to show you that. And I also wanted to stress before we round this out, really how much we haven't shown you. One of the things that we didn't get a chance to look at was doing PHI handling in place. Once you do that, very intensive logging starts to occur. LabKey will log when you have your data protected by PHI wards, if you like. It will tell you who looked at what data grids, which participant IDs, and it'll give you a report and query and give you reports on that activity. It also will allow you to do very more strict account management. So you can tell people, you can tell the server, I want this person's account into my server to last only a few days, or to expire after a few days, or to expire after they've been inactive for a few times, etc. It also lets you limit login attempts. You can also do electronic signatures on your data as part of compliance. And so that's just a few of the things we didn't look at. And also, I wanted to point you at some of the next steps. You know, if this kind of sparked your imagination, what should you do to keep going? Well, first off, I highly recommend to get your own trial server. All of my work was done on a trial server. I built a trial server to do all this. You can do the same, go through our tutorials, and you can have much the same experience that you saw me go through today. Basically, what I did is I went through a tutorial live today. I went through the set up a new study tutorial. You can do the same, get hands-on with this. Also, we are going to be doing these LabKey Academies every 2 months, so watch for the next one. It's going to come up. I'm not sure what we're going to do, maybe assays. Will you tell us? You know, what would you love to see in 8 weeks? Also, there's a much broader set of features we haven't looked at. We have a survey module that lets you do EDC directly on the server. If you have REDCap data, you can pull it into your studies. We didn't even touch on specimen integration. And we didn't even touch on assay data integration. That's all stuff that's just right there. We did take a tiny little look at the LabKey API with R. But again, there's just way more to do, way more coming as you keep going. Also, we have a survey. If you want to give us some recommendations or what you want to see next, please do this survey. I'm going to put the link in the chat. And right here. So I've got 2 links for you in that chat. One to take the survey and one to look at the site I just built. And any other questions that I should address, Jason?
I think furiously behind the scenes here, we've been addressing most of the questions. So
great.
The ones that were more. Sorry.
Yeah, great. And did you have any parting words, Jason, before we round this out?
Just a couple of things, really, to iterate what you just said, Steve. So this is a new training series. We welcome your feedback. We want your input and some guidance as to what other topic areas you would like us to cover on a bi-monthly basis. My email address is there, jasonl@LabKey. Please feel free to send comments, feedback through to me, and we'll address thereafter. We do have another webinar, which is planned on July the 14th. Most of you will have known that we released LabKey Sample Manager earlier this year. So dedicated specifically for creating samples, tracking the processing of samples, chain of custody, and the ability to integrate multiple assay data at the sample level. So a very neat, intuitive UI that's available and included for no extra charge in all our premium versions of LabKey Server. So we'll be covering that on the 14th of July, so please register for that on the website. Also, each year we do have a LabKey user conference. We have one based in Seattle, which is 7th to the 9th of October, and one in the springtime in Europe. Now, for obvious reasons, with the pandemic, we've had to cancel those in-person conferences this year. But we just wanted to let you know that for the Seattle replacement, we will be doing an online version of the user conference. The dates are the same, 7th to 9th of October. It's free for this year, so we welcome your participation. It's a great way to hear firsthand presentations from users of LabKey, both Community Edition and premium versions, and to get insight into how they themselves are applying the software to solve their own research challenges around the world. Really insightful, lots of value to be gained from that. We will also, of course, be providing updates to the platform, new features, new functions, etc. So all this will be published under the events section of our website, so I invite you to take a look at that. But please keep us informed, communicate with us, let us know via the email address there. Thank you for joining, and we appreciate it. Thank you, Steve, for sharing your knowledge and your expertise, and we look forward to welcoming you back to our next webinar series. Thank you for joining.
Great. Thanks again, everybody. And again, I really recommend people look at that LabKey user conference. This is a huge resource. I'm going to be doing 8 hours of these kind of presentations. It's really just the best way in to LabKey server to get your head around it, because it's really just such a huge piece of software with so many features in it that we haven't even touched on. Got 3 minutes left. Any questions that I should just try to jump on, Jason?
Just scrolling through these very quickly. There's lots of thank yous.
Great.
Topic of suggestion for next time is integration of instrument data. So assay component.
Yeah, we'll do that either directly, or if we do samples, that will include some assay data, integration as well. And this is the first time we've ever done this in an hour-and-a-half format. I think I was a little bit ambitious. I had to skip some stuff. So, if you have any questions, you're welcome to, ask them at the next one as well.
I also think, Steve, it's worth mentioning that, 99% of what you show today is in the community edition.
Oh,
so
I'm really glad you mentioned that, Jason. Yes. The only feature that I showed that was a premium edition feature was that file watcher feature.
Yeah.
So this was basically a demonstration of community edition features, which are free. It's amazing how much you can do with the community edition, just to stress that. Okay, well, thank you very much. I'll sign off a bit early and again, thank you much.
Take care, everyone. Thank you.
Bye-bye.